Hey, welcome everybody. We are now to experiment 7 in our series of CRISPR experiments. In this experiment, what we're going to do is we've already gone through and we've made our mutants, we've screened them, the DNA sequencing came back, and we now have grown up, the TAs have grown up cells for you guys to purify. So they've gone through and one thing they've grown up is a strain called BW1418, and this strain is what we're considering to be our wild type, even though it doesn't have the presence of the pho-8 gene. Pho-8 gene is another gene that makes an alkaline phosphatase. It's vacuolar, and this is not the one we're interested in. So Dr. Wasco created a strain that deleted that gene. Now we have it, so this strain will produce just pho-13. The only alkaline phosphatase it produces is pho-13. So we have that wild type one, and in this case, what I'm going to do here is I'm going to show you how to do purification using the 1418, which is the pho-8 mutant. And we'll start that off. And also, this whole video I'm going to go through and I'm going to just do the control, while in your experiment you're going to be doing the control as well as your mutants. And we want you to do both. We need you to do both so that whenever you do experiments to one, we can see what happens for the other one, and we can compare them, so we'll be able to say if the activity is higher, lower, or what the differences are. But to be able to control for that, we want to go ahead and use the original strain that we made the mutation in. So the first step in here is we want to add 1 milliliter of cell-lytic Y per gram of wet weight cells. On your tube that we'll be giving you, you'll have information on here. It'll have a tare weight, it'll have how much was made, and then it might even have how much total the pellet might be. So there's 1.3, in this case there's 1.32 grams of yeast cells in this tube, and it was frozen, and I've just now got it thawed. And now I will add 1.3 milliliters of cell-lytic Y. So, and I'll do that as 0.66 twice. Add the first one, add the second one, and now we need to mix it up. Your pellet, this pellet was kind of soft, so it dissolved. As it thawed out, it kind of dissolved. So just, it depends on what I do is I pipette up, and I would just go ahead and just kind of pipette down on the pellet to dissolve it up. And here it looks pretty good. Here, and now I want you to do this to both the wild type strain, which is the pho-13 strain, and you'll also do it to your mutant. And here you just go ahead and just kind of stir it up and just let it sit at room temperature for half an hour. And I'll be back in about a half an hour, and we'll walk you through the next steps. See you back in half an hour. All right, I'm back from the centrifuge. We have this tube here, and then this is what we centrifuge. And you see here, the white stuff, this white kind of beige kind of stuff is the pellet. So that's cell debris and other things we're not interested in. And this clear solution up top here is what we refer to as lysate. So in the protocol, this is what we call lysate. So we gently take it out. And you try to take it out, try to minimize, try not to disturb this pellet at all. So that this pellet, now we can throw that away. And now we have this solution that I refer to as lysate. The first thing we need to do is to remove 100 microliters out of this lysate, put it into another tube, and this we're going to save for experiments we're going to do next week in class. So this 100 microliter sample is saved for next week. Now we have about a milliliter of lysate here. So what we'll do is we're going to do chromatography. We're going to use this little column. Note there's about 2 milliliters of this DEAE cellulose present here in the column. And what we're going to do is we're going to pipette out the lysate. And what I'll do is I'll be putting it in here, and I'll be just slowly adding the lysate to the top of the column to minimize the disturbance to the top of the column. Will I be able to do it without disturbing it completely? Probably not, but try to do it minimally. So just kind of add it kind of slowly, and just kind of slowly add it. Much like you did for the gel filtration step in an earlier experiment. So we added the first part. Now let's add the second part, this residual in the tube. Here we'll go ahead and add a little bit more. So we added about 1.5 ml. So the next step on this procedure is they actually recommend adding 1.5 milliliters of pH 9 buffer to this. So we have some pH 9. You'll have it in a smaller container. I'll just do it out of this larger container. And I will slowly, it's set at 0.75, and so I'll do this twice. Just slowly add this to the top of the solution, trying to minimize any disturbance. Okay, so it's been added to the top. Now, what I'll do is, I've already got these tubes. What I did is I sat down and before I started doing this chromatography step, during the half an hour of lysing the cells, I went and labeled the cells. So what I did is I have 3 2 ml tubes labeled load, and I do that load 1, 2, and 3. Then I have a wash 1, 2, 3, 4, and 5. I have a 0.1 molar 1, 2, 3, 4, 5, 0.3 1 through 5, 0.6 1 through 5. And I have those tubes already isolated ahead of time, and those will correspond to the buffers that we'll be using. So what I want to do is I want to collect all this flow through. It's kind of, in case something happens and everything flows through, you have this material so you can potentially use it again. You won't lose it. So what we'll do is here, we'll remove the cap here on the bottom. Oh, something I didn't tell you is that off camera and before I started doing this, I ran 12 milliliters of buffer through this column to make sure it was equilibrated and ready to work. So this column's already been equilibrated. I washed it. The protocol says to do 6 mils. Because I was doing other things, I went ahead and put 12. I filled this whole thing up and equilibrated the column. You'll be doing this twice, and the protocol tells you to add 6 milliliters and wash it through after you've done this step. So the first thing we'll do is on this load column, we'll remove this little cap and start collecting the material as it comes out. This isn't terribly exciting, but we go ahead, and the larger the volume is, the faster it'll drip. As it gets closer and closer to the bottom, it'll drip slower and slower. But this will be a little faster, a little bit better, easier to do than the size exclusion experiment you guys did earlier in the semester. I went ahead and put 3 tubes here, known as 6 milliliters. I don't think we're going to need this many, but we'll go ahead, and I always like to have an extra tubes present, more than I will need. So you see this one is getting close towards the top. So what I'll do is kind of switch hands, and as this one drips and fills in the top, I will switch and start collecting again. I go ahead and cap the tubes and put them off to the side. We'll be using these tubes next week for next week's experiment. And as you notice here, the solution's getting lower and lower and lower. And what I'm doing is I'm watching it as it gets close to all the liquid being gone. I'm going to put this little cap back on. Getting very close. Notice that it's not dripping quite as fast as it was, because the total amount of buffer on the top will determine that flow rate. Okay, it looks like it's basically stopping to drip, so we'll go ahead and put this cap back on. We don't want the column to be able to drip long enough to go completely dry, because that will damage your sample. So in this case, I had 3 tubes reserved, and I only used 2 of them, so I'll put this 1 tube off to the side here. And so the next step is to add wash buffer to the top of this column. And I have the wash buffer sitting in one of these tubes. And for the wash, it's 10 milliliters. And unfortunately, the easiest way to do it is not necessarily fun to do, but we'll put 10 1 milliliter aliquots on the top of the column. Again, we'll go ahead and do it very, very slowly. The first few we have to put on very, very slowly so we don't disturb the top of the column. But as you get closer and closer to 10 milliliters, you can actually start adding it faster and faster because it won't disturb the top of the column. But the first probably 3 milliliters, try to add really slowly, because the vast majority of your protein will probably be bound to the very top part of the column. It's 2. That's the first one. This is the second mill. So keep track of how many milliliters you've added. This is the third mill. I can probably add a little bit faster here because it probably won't disturb the top as much, but I'll still go at it kind of slowly just to make sure that we minimize the disturbance of the column itself. With each of these stages, we'll be doing several of these. We'll be doing 3 more stages of adding adding buffer. Okay, here's the 10th milliliter. Put the cap back on the buffer. And now this is a buffer that's kind of just washing all the material off and re-equilibrates the column, getting it ready for the elution step. And what I've done is I've labeled these tubes wash 1 through 5. And there's about 2 mil tubes. We've added 10 milliliters of buffer, so we just start collecting them. It starts off kind of fast, and we'll slow down, just like the last time. What we're doing is we're basically washing these columns with 5 column volumes of buffer. So the column volume here is 2 milliliters, and we're adding 10 milliliters, which is 5 column volumes. And that should be more than enough to completely elute the peak. Right now, this will re-equilibrate the column, wash it, and then in future steps for elution, it makes sure that the whole sample will elute from the column. So what I've also done is I've organized the tubes in line. So what I do, as soon as I've collected one, they wind up going 1 slot further down the rack, so I see a gap of the current tubes, and then I know where the next tube is going to be. Okay, the column's almost dry, so we'll put the cap back on it. And now we have 5 tubes of the wash. It's labeled I always label them wash 1, 2, 3, 4, 5. I don't know if you can read that. So the next step will be doing 10 milliliters of the 0.1 molar wash. I'll be back in a minute. All right, now it's time to go ahead and do start doing the elution process. In ion exchange columns, you elute protein by adding increasing amounts of salt. So the proteins will have a certain amount of charge, and it requires a certain amount of salt to remove them from the column. So we're going to start here is we're going to start doing this sort of step elution approach. And the first step we're going to use is to use 0.1 molar sodium chloride in the same buffer. So it's pH 8.1 tris buffer, 20 millimolar tris buffer with 0.1 molar salt. So we'll go ahead and again, we're going to be adding 10 milliliters, which is 5 column volumes. And we'll do the same thing we did the last time of just kind of slowly adding it to minimize the disturbance to the top of the column. Okay, so I have now added 10 milliliters of 0.1 molar salt. I'll get my first tube out. I have labeled 0.1-1, so I'll just go ahead and take this cap off and start collecting. So I'll go through the same thing I did in the past step, is just go ahead and fill this one up, fill each tube up as we go. Small little columns actually elute relatively quickly, which is kind of nice, and that's all we really need. We don't have a lot of protein in these samples, so we can use these small little columns and batch elute them and actually go kind of fast. This makes it so that you can easily run the column twice in one lab session. Okay, it's almost dry, so it's time to cap it. We'll put the cap on it and seal this tube up. So the next step is to do the same thing again, and we're going to elute with 0.3 molar sodium chloride. So we have them they'll be in your bench. You'll probably be in tubes sort of like these. So we'll start adding. Three-tenths molar salt. Again, same thing every single time. The first 2 to 3 milliliters you need to add very slowly to minimize disturbance to the top of the column. I know as you do this, you probably want to do it faster and faster, and it's harder and harder to do it slowly each time, but it's best if you do. I've added the point. Now at this point I've added 10 milliliters of 0.3 molar sodium chloride, and I'll get my first tube labeled 0.3, number 1. So move the cap and then immediately put this underneath it. So start collecting again. So we do this 4 different times, well 5 different times including the initial load column. And as always, the first one goes relatively quickly. The second one takes longer. Each subsequent one takes a little bit longer. But it doesn't take too long to do this. Okay, now we just collected the last of the 0.3 molar salt. So the next step is to go ahead and add 0.6 molar salt. And this, we'll do the same steps we did for all the previous ones. Slowly add the buffer to the top of the column. So I've now added the 10 milliliters, the 5 column volumes of 0.6 molar salt, and I'll start collecting. So here's the first tube. Start collecting. Okay, now I've collected and I put the cap on. Collected the 10 milliliters 5 times 2 of the 0.6 molar. Now what you need to do is you need to go ahead and get the column ready again to be able to use again. But go ahead and add 12 milliliters of the 20 millimolar tris buffer to the top of the column, re-equilibrate it, and then you can add the lysate and start back over, I think it's step 2 of your protocol. Add the lysate in, add a little bit of the pH 9 buffer in there, and then go ahead and collect that load and go through the same process again, either with your mutant or the wild type. You can do them in either order, it doesn't matter, but just as long as you do both of them the same way. Hopefully that helps, and we'll see you in the lab. Bye.